en resumidas cuentas, porque su currículum es muy extenso. Ella obtuvo su doctorado en química de la Universidad Autónoma de Madrid en 2011. Bueno, puedo decir en inglés ahora que ya empecé. She developed her PhD at the Instituto de Ciencias de Materiales de Madrid under supervision of Professor Gutiérrez Puebla and Dr. Snesko. In 2012, she joined the Stockholm University of Sweden as postdoc researcher with the Vercelli Center Excellence on Porous Materials. 2014, she moved to the Argonne National Lab in US as a senior postdoc. In 2018, she started her independent career in UAM, so Autónoma de Madrid, through the Talent Grant from the Community of Madrid for more than 300,000 euros, a lot of money. Since March 2019, she's a member of the Condensed Matter Physics Center, IFIMAC. Ana is also a recipient of the prize fellowship Ramon and Cajal since November 2020. She's currently leading a research group at the UAM composed of 2 postdocs, 2 PhD students, and 1 master student. So the group is founded for several projects from Spain, Spanish Ministry of Science and Condensed Matter Physics Centers, and she has over 660 publications with 32H index. So we're very happy to have her here. Estamos muy contentos de tenerte aquí, Ana, y bueno, la pantalla es tuya, puedes comenzar tu charla y bienvenida. ¿Nos escuchás, Ana? Nosotros no te estamos escuchando. Vemos la pantalla, perfecto, sí, pero no te estamos escuchando si estás hablando. Tal vez... voy a chequear si...
Ahora, ¿no?
Ahora sí, perfecto.
Vale.
Ahora te escuchamos, Ana, perfecto.
Perfecto.
Perdón. Ahora
Cosas
sí.
directo.
Sí, no hay problema, estamos preparados.
Vale.
Bueno, bienvenida de nuevo, gracias por estar y bueno, estamos ansiosos por escuchar tu presentación.
Vale, muchas gracias. So thank you very much for, first of all, to the organizers to give me the opportunity to share with you today part of my research, which is about metal organic frameworks for catalysis. And in the next minutes, we're going to be talking about a very fundamental question, which is how can we explore the atomic structures of metal organic frameworks without crystals, okay? So to do so, I'm going to explain you a new technique, which is called pair distribution function or PDF analysis, by showing you different examples of the research that we have done in the last years. So, so the question in this talk is really, how can we characterize in the best possible way, the structure of a material, of a metal organic framework? Okay, so for me the question here is, at which scale are you interested in? Because if we follow the most conventional approach, usually we focus in the long range order, so periodicities beyond 30 angstroms. So this is a good approach if you are interested in looking into the topology or the connectivity of your framework, which are the kind of pores that you have, the size, the shape. That's a correct approach. However, if we manage to really zoom into the structure and study the local structure, we are going to be able to determine the periodicity of chemical bonds, molecular interactions, and local defects. And in my opinion, this is key to harness localized properties such as catalysis and adsorption in metal organic frameworks. So how can we do that? How can we study the local structure of metal organic frameworks? So one possible approach is to perform high energy X-ray total scattering experiments. So in these experiments, the characteristic of this experiment is that we can combine, or we can collect simultaneously, Bragg diffraction, which is related to the long-range order, with diffuse scattering, which is related to the short-range order. And this is very important, okay? This is something I want to stress. In the same experiment, we are going to analyze Bragg diffraction and diffuse scattering. The Bragg diffraction is linked to the average structure, so we can determine average atomic positions, cell parameters, occupancies, okay? But if we study the diffuse scattering, so this is usually what in our diffraction pattern we are going to describe as a background. So, well, if we manage to study in detail this background, we can identify two-body correlations, which in my material is linked to chemical bonds, distortions, interactions, okay? So the way to analyze these two pieces of information together is by doing the pair distribution function analysis or the PDF analysis, okay? Which at the end of the day is a weighted histogram of all the atom distances that you have in your sample. So your sample can be anything you have in your mind. So it can be a liquid, can be an amorphous material, and can be also a crystalline material like a metal organic framework in which we suspect that the local structure can be different, okay? Because we do some activations, some chemical modifications, for instance. So in here in this slide, on the bottom side of the slide, you can see which is more or less the processing of the data. So this is the raw data, the scattering pattern. And then after applying some corrections and the Fourier transform of this data, we obtain the PDF profile. This is the PDF profile of a metal organic framework, okay? So one of the beautiful things of this technique, in my opinion, is that it's very intuitive. So the signals here are directly atom-atom correlations or distances in your material. So this is very interesting because now, depending where are we, so if we are at the short radial values, we are getting information about chemical bonds, the coordination sphere, so something very important for a chemist. And then if we manage to get information a bit farther away, so here we get information about intramolecular interactions, which is also very important if we want to study catalysis, adsorption, etc. So now in the next minutes, what I'm gonna do is just to walk you through different examples of the work that we have been doing in the last years to show you different examples of what we can do with PDF analysis to better study metal organic frameworks. And I'm going to start by discussing this paper. This is a work from my postdoctoral period with Professor Chapman at the Argonne National Lab. And it's a work that I like it very much because I think in this work, so this work was critical to demonstrate to the community that if we really want to understand how is the local structure of the metal organic framework, the better approach is to use PDF analysis, okay? So with Karina, what we were doing is to use this is a zirconium MOF, the NU1000 from Northwestern University, and the zirconium MOF, in which we were doing different chemical modifications for catalysis. So here you have detail of the zirconia cluster. So us as the characterization team, our question was, okay, what is the local structure? What is the precise, let's say the precise structure of the zirconio cluster under conditions which are relevant for chemical modifications, for activation before catalysis, okay? And what we did is, well, I cannot pass the slide.
Are you having problems to
I'm
pass
having
the slides?
problems. Yeah, I cannot pass.
Maybe stop sharing, stop presenting whole screen and then go back again. Maybe that will help.
Let me.
If you click on the just click escape. Maybe
I'm gonna stop sharing then. Yeah, just to maybe to reset.
Yeah, maybe that's already stuff that happens.
Okay, let's try.
Yeah, it was working fine, right? You were able to.
Yeah, at some point just.
Okay, now we're seeing your screen.
No, I can't. So
you can. So let's try. There should be nothing different from our side, so it should be the same. So maybe if you close the PowerPoint and open it again in your computer, maybe something's wrong with that.
Let me try.
Yeah, now we're seeing not in presenting mode. So now if you go to
Yeah, I'm just gonna try. Let's see. Okay,
images.
now it's working.
Yeah, okay, perfect.
Okay, sorry for this.
No, no worries. It's fine.
So we were discussing about NE 1000. Okay, is this zirconia metal organic framework, and that was a collaborative work with professors Farha and Hab from Northwestern University. So what we did at the beamline is to take this MOF, to put it at the beamline, and then to perform an in situ PDF experiment in which what we did is to change the temperature from 50 degrees until 350, and then to collect, to do fast PDF data collection, okay, to follow any potential structural change. And this is the data that we got. And here, please, I want you to focus here in the middle of the slide, and I'm just going to explain slowly what kind of information we got from this PDF experiment. So this, first of all, I just want to explain, this is a small region of the total PDF profile. So I just focused in between 1.5 angstroms and 4.5, because this is the region to explain the coordination environment of the zirconia cluster, okay? So we have more information, but in order to explain this case, I just focused in this region of the PDF profile. So the first signal that we get, around 2.2 angstroms, this is associated to the zirconium oxygen chemical bonds, okay? So in this PDF experiment, and here we have in blue the low temperatures and in red the high temperatures. So what we saw is that by heating, we have a decrease in intensity in the signal, which is related to the loss of water molecules in the coordination sphere of zirconium. So this was kind of expected because we knew that we could dehydrate the cluster. However, let's look at the second signal. The second signal is related to the zirconium distances. So these guys here, okay, within the octahedron, within the cluster. And this result was completely unexpected for us. So let me walk you through the experiment. So at room 50 degrees room temperature, we start here. So we have a single signal around 3.5 angstroms, and that makes sense because this is the distance zirconium in the regular octahedron. Okay, so we know that for this structure, if we get the structure from single crystal data, this is the distance that we should have. However, as soon as we increase the temperature, we see how the intensity of this guy decreases, and then we have the appearance of two new signals, one at shorter distances and the other at longer distances. And it's important to explain here that the intensity of these two signals is exactly the same intensity that you have here. So that means that we are not decomposing the cluster, okay? This means that we have local distortion in which the symmetry of the cluster is reduced. So we go from something that is very regular to something that is a bit more irregular. And this is why we get two different distances. So simultaneously, we were collecting diffraction data. This is important because we could see how this local distortion is occurring without disrupting the framework connectivity. So in other words, the framework is not collapsing, okay? So just think about it, because without PDF, we would have missed completely this local distortion in the cluster, okay? So the next thing we did is to go a little bit further and then to perform quantitative analysis of this PDF data that I showed you 2 slides before. And this is a complex graph, so it needs a little bit more of explanation, okay? So first of all, let me explain. So in the y-axis, we have normalized peak area, and the value is 12. And it's 12 because my reference is the octahedron, okay? So an octahedron has 12 edges. So we start the experiment 50 degrees. What I have is 12 distances, which are the same, 3.5 angstroms. So we are fine. And then at some point, which is close to 150 to 100 degrees, things happen and the intensity of the signal decreases. And then we have the appearance of two new distances, the short one, 3.3, and the long one, 3.7. But thanks to this quantitative analysis, we can determine the exact number of distances that we have. So after the heating process, we can say that the new structure has 8 short distances and 4 long distances, okay? So with this experimental piece of information, we went to our colleagues, the theoreticians in University of Minnesota, and what they tried to do is to simulate possible structures that could explain the experimental data. So the first thing we try is to just remove all the water molecules and then let the structure optimize, okay, and then see what was happening. And that wasn't the answer, because the structure that they got contained all the zirconium distances contracted, and this is not what we were seeing in the PDF. So remember that we see short and long distances. So we were, of course, not happy, none of us, with this explanation. And in that case, what we did is to use ab initio molecular dynamic simulations, which is very useful in this kind of studies when you want to reach a little bit more complex structural mechanisms. So by applying ab initio molecular dynamic simulations, we could catch actually, yes, that first we have the dehydration process. So we remove some water molecules, the water molecules in the cluster, but afterwards we have a change in the coordination mode of the hydroxyl groups. And this is key to understand the structure that we have at the end of the heating process, which is this one here, what I call the distorted cluster. So this structure agrees very well with the information that we got from the PDF. So we have 8 short distances and 4 long distances. And here what I want to show you is the following. So what we did is to take the symmetric structure. So this is the symmetric structure of the cluster at room temperature. And then this is my distorted structure at the end of my local the structural transition. So what we do is to simulate which is the fingerprint in the PDF signal, okay? So if you go from here till then, this is the fingerprint in the PDF. And then we were shocked when we checked back in the literature, so things that are well known for bulk zirconia, because zirconia has many crystalline phases. So we took the crystal phase of the cubic zirconia and then the monoclinic, and then we simulated the same local distortion going from cubic to the monoclinic, and then we were puzzled when we saw that actually the phenomenon is exactly the same. So one is happening in the bulk, so all the structure is changing, and in metal organic frameworks, it's happening in a very localized way in the nanoscale, okay? But more interestingly, what is very interesting is the things that are different in between the bulk and the nanostructure in the MOF. So what we learned from this work is that the transition temperature is substantially depressed at the nanoscale. So look, what is the temperature that I need to reach for the zirconia to go from the monoclinic to the cubic, which is 1000 degrees Celsius, is very high. And the local transition that we determined, it was happening around 120, 150 degrees, which is a temperature that I think all of us can agree that is very relevant for any treatment that we do with MOFs in our laboratory. So I'm just thinking about mild activation processes or post-synthetic modifications. So just consider that you might have local transitions in your material as well, okay? And then another thing which is, of course, of relevance is that the order of the transition is reversed. So for zirconia, we know that the thermodynamically stable phase is the monoclinic, okay? And then we go to the cubic one at high temperatures. While in the case of the metal organic framework, we have the regular structure stabilized at room temperature, most likely because of confinement effects. And then when we heat up and remove some water molecules, we get the distorted conformation of the cluster. So this, beyond being like answering ourselves a very nice fundamental question, it was very important for not only for our project, but for many other projects in this network. So because we could demonstrate that the NE1000, which is a zirconia MOF, which are usually they are well known as a very robust metal organic frameworks, can tolerate substantial number of local distortions or defects, okay? And more importantly, these defects, they have an effect in catalytic properties. So the node distortion may explain the higher activity of this material for hydrolytic degradation of chemical warfare agents after dehydration. So this is something that we couldn't explain before this work. So we knew that if we dehydrated the material, that one was 20 times faster or more active for the catalytic dehydration of chemical warfare agents, but after determining the local structure, we couldn't explain, okay? So this is just to stress how important it is to know how atoms are connected, because if we don't know that, we cannot really tune and tailor the properties of metal organic frameworks. So after that, we of course explored other systems, okay? So this is NE1000 in which we managed to deposit Caperoxo clusters. So here you see the location of the clusters, which is they are located in between zirconia nodes, okay? Here is my PDF data to show that indeed we have the corresponding oxygen zirconium distances. And what I want to explain you is the results from in situ diffraction and PDF experiments. So I'm going to start by the in situ diffraction experiment. So this is the data from in situ diffraction, and we explore conditions which are relevant for the activation process in this material, which consists of treatment with hydrogen at 200 degrees Celsius for 2 hours. So in this graph, I have summarized all the information. So here we start at room temperature. Okay, so this is my cell parameters at room temperature. And then as soon as I increase the temperature up to 200, what is happening is that there is a significant expansion of the C parameter in the structure. Okay. More interestingly, if we expose now, we switch helium by hydrogen, we start the reduction process, many things happen. You see that there is a significant contraction of the A parameter and then an expansion of the C parameter. So in order to understand mostly this region, okay, we were very intrigued by this region here during the activation process. We perform as well in situ PDF analysis, so following exactly the same conditions, because we were wondering if something was happening to the Caperoxo clusters that could explain these major changes in the cell parameters. And indeed it was. So here I have plotted 4 representative PDF data. Okay, so we have plenty, but these are the most representative. So we start, this is my pristine material. So the material with the Caperoxo clusters before any treatment. And then it's nice to see that as soon as you expose the material with hydrogen at 200 degrees, like it's very fast, okay? So we have the appearance of this signal here, which corresponds to small clusters of copper zero, okay? Really small clusters. So, and then also the formation of copper 1+ oxide and nanoparticles. And actually these copper oxide nanoparticles, 1+, what happens is that when we increase the treatment, the reduction treatment over the time, the size of these nanoparticles increase, okay? And then after exposure to oxygen, and this we did it just to understand better the deactivation process of the catalyst, what happens is that we lose completely the copper zero clusters, which indeed are the active species in the material. So this is just to show you how powerful the PDF technique, so we can identify which are the active species, and we can even distinguish in between different oxidation states if they have different crystalline structures, okay? So later on in Madrid, we were working with another zirconia metal organic framework, the ArMOF 808, and this is a work done by a former postdoc in my group, Celia Castillo Blas, who is now working at University of Cambridge with Thomas Bennett, continuing doing PDF because she liked it so much that she wanted to learn a little bit more. So Celia did an amazing work because she managed actually to introduce iron single site in this metal organic framework. And you know, iron is a very tricky metal because iron, what it likes is to form nanoparticles and to aggregate. So it's really difficult to stabilize single sites. So this is just diffraction data to show you that the materials were crystalline after the incorporation of different amounts of iron. And because what we really want is to understand better the structure of the active sites, what we did is to propose different, to run calculations and to propose two different configurations, structural models for the cluster. And the results are here, okay? So there were two possible conformations for these iron clusters deposited on the MOF 808. So one possibility, this was the most, let's say, the conventional approach, is to suggest that the cluster, the iron cluster, is coordinated to one zirconium node, so and then is pointing towards the big pores of the materials, okay? And the other possibility is here. So the iron cluster is actually acting as a bridge in between zirconia nodes. So the calculations were saying that this conformation here is thermodynamically more stable. And actually our PDF experiments, by looking at the iron-iron distances, which are difference between this conformation and the other, we're confirming that the most plausible configuration of the iron cluster in the MOF 808 is actually this one here, okay? So that's very interesting, because having this kind of heterobimetallic nanostructure, so being possible to stabilize iron at the nanoscale at this level, so it's very interesting for catalytic transformations, because we can avoid aggregation processes. Actually, we demonstrated this by testing the material in femton reactions, which are, well, is a transformation for degrading organic pollutants in water. So this material actually is able to stabilize the iron active species in this way, okay? So we wouldn't have been possible to identify this structure without PDF, because of course we don't get single crystals, okay? So beyond metal organic frameworks, let me just walk you briefly on covalent organic frameworks as well, because they have many things in common. So this is a work done by my PhD student, Nacho, who did also like an amazing work. So this is the covalent organic framework that we were working. So it's a covalent organic framework based in imine linkages. And in this work, we were comparing two synthetic methods. In one, so here on one hand is a method in which we know that the material is defect-free, so it's very crystalline, we don't have defects. And then we developed another method in which the material contains a lot of defects. So what is a defect in this material? You can see it here. So what I mean by a defect is a local defect in which where the imine linkage has been hydrolyzed, okay? And then we have, instead of imine, we have a carbonyl group, haldehyde, and amine, okay? So at this point, what we really wanted to see is what is the conformation of this defect, because you can have a cis or a trans conformation by looking at the orientation of your oxygen and your nitrogen groups, okay? You have here the carbonyl group from the haldehyde, you have the amine, and they can just be like pointing towards different pores or pointing towards the same pore. So this is very difficult to answer if you don't apply theoretical calculations. So we did, and we could demonstrate that the trans conformation is favored by 5 kilocalmole, which is significant. So this is the structure that we most likely we have in the materials with defects. And once we knew this, what we did is the next step is to use this defect to incorporate catalytic metal sites, okay? So in that case, we used palladium because we were interested in performing Suzuki-Miyaura carbon-carbon coupling. So this is the palladium. This is just part of the diffraction, because we always have to show that we preserve the long range order, so the connectivity. But for us, the important thing is to understand how palladium is coordinated to the material, okay? And we explored two different possibilities. One possibility is here. So that one here, in which my palladium dichloride, by the way, so is coordinated to two imine linkages, okay? So it's connected to what we call the pristine linkage. The other possibility is that the palladium actually is going to the defect. So we have a defect in which we have a carbonyl, imine, and so this imine from the previous layer, okay? And then the amine here. So the possibility is that the palladium can coordinate to imine amine. Okay, it's very subtle, but this makes a difference, okay? So our calculations were showing us that actually the structure that is more favored is that one here. So that was very interesting because it means that as soon as you have defects in your material, palladium is gonna go there. So palladium is gonna like better to be located in the defects, okay? So we tested our materials for the Suzuki-Miyaura coupling just to prove that actually this palladium is accessible, okay? It's also worth to mention that the active species are palladium nanoparticles. So we start from palladium 2+, which is a precursor, and then we form palladium nanoparticles. This is a well-known mechanism in this kind of catalysis. And the next thing we did is to perform, we went to synchrotron, to the Soleil synchrotron in Paris. We performed X-ray absorption spectroscopy because this is very powerful to understand better oxidation states, coordination spheres, okay? So here is the data of two materials. So this is my material without defects. This is what we call the late metallation, and the material with a huge amount of defects in the structure. So we know that here palladium is going to be binding to imine, okay? And here is going to be binding to the defect, so imine, amine. So look at the signal corresponding to the palladium-palladium distances. We can see how for the material with high amount of defects, this intensity here is much lower. And that's important because that means the palladium nanoparticles are smaller. So it means that we have less aggregation after catalysis. And this is a very good result, okay? Because this is everything you want when you have a catalyst, palladium catalyst in particular, okay? Because it's very prone to aggregate and then you lose activity. And of course, when we saw this result, we thought, okay, this is a very good candidate for PDF analysis as well. So we performed PDF analysis of the two materials before and after catalysis. And well, I want you to draw your attention here in this slide here. So in this graph here, sorry. So if we compare the material before catalysis, the one without defects and the one with defects, the early metallation, we can see how we have more signals in the short range. So it means that the palladium is actually, let's say, better localized. So it has a better ordered in the material with defects. So the connectivity is better. So the coordination environment is more ordered, okay? And after catalysis, we see that we have less signals or less intensity here at high R values, which means that the particle size of the palladium nanoparticles is also smaller, okay? So that was in very good agreement with the XABS analysis. But something we can do with PDF that we can't do with XABS is to estimate which is the precise size of the crystallites in your material. So you can get your PDF, and it's very similar for those people who are familiar with diffraction with Rydberg analysis. So very is the same philosophy actually. So we did this kind of Rydberg analysis to determine which is the particle size, not particle, the crystallite size, which is about 3-4 nanometers. But more interestingly, when we subtract the signal of the nanoparticles, we could identify the formation of this palladium bromide, which has a very distinctive chemical bond distance, okay, 2.4. And so thanks to this analysis, we could identify that in the material, actually, we have also trapped this catalytic intermediate in which the chloride ligands, which are very labile, are replaced by bromide groups, which are formed during the catalytic cycle. So that was a really nice result, because I think it's the first time that someone is reporting the localization, so the determination of these reaction intermediates in a covalent organic framework. Okay. So what I want you to really tell you about this work and what we do with PDF is that the defects are everywhere. So even crystalline materials as metal organic frameworks, they have lots of defects, and defects are very active. So that the structures of catalytic metal sites within porous materials are richer and more dynamic than we thought. And if we don't have the proper tools to look at it, we are going to miss it, okay? And that we need to provide precise knowledge of the local structure to better understand the catalytic performance of metal organic frameworks. So to finish, I would like to thank to my amazing group in Madrid, with the people who actually do the real job, the real work, for also to the funding agencies to give me the opportunity to do the research that I want, and to all of you for your attention. So now I'll be more than happy to take any question that you might have. So thank you.
Thanks, Ana. It was a really nice presentation with a lot of concepts and ideas that will be surely appreciated by our audience. And I will start because I have some curiosity about the first slides you present, and then we have maybe 2 more questions from our audience. I was wondering, which this is interesting work you presented on the first PDF application you show with zirconium clusters and variation of distance in let's say zirconium oxygen and zirconium.
Yeah.
So you show nicely that the structure is not altered by the XRDs you presented, but I was wondering, what about if you have measured, or if you haven't, I mean, maybe it's something to be done, but the porosity of the system, it is modified by this, let's say, alteration of this distortion in zirconium distances, or is maintained?
Not substantially, no. We didn't see any significant change that you might suspect, okay, this is coming from the local distortion. Although it's something that, to be honest, we didn't look into it much. So maybe if someone is interested, it's something interesting to see. If but yeah, nothing really significant, let's say. Yeah.
Let's say, well, because the heterogeneous catalysis activity should be influenced by maybe the surface area porosity, and if it's altered, then so I was wondering.
That's a very good point. Yeah.
Okay, thank you about that. We have some questions from the audience. Here's Marcelo Ciolin, a local expert on XRD and synchrotron radiation sources. He wants to congratulate you on your nice and instructive talk. And he would like to know if you have also performed XAF or XANES experiments or anomalous PDF experiments in your samples, and if you have any comment on relative advantages or complementarities between PDF and these other techniques.
Yeah, that's always the question, actually. So so they are complementary techniques, actually. So in my work, we typically use, we combine PDF with XAF. So XAFs, XANES. So XANES is giving you information about oxidation states, geometries. So that's very sensitive to identify geometries. The point, if you really want to determine local structures, in my opinion, XAFs is not the best way to go with metal organic frameworks, because you may have, so typically with zirconium MOFs, we don't see correlations, so we don't see signals beyond, with XAFs, beyond 3 angstroms. So that's a pity, because if you want to study the structure of a cluster, you don't have this information. And this is typically because, depending on the energy you work, you can have a lot of absorption of photons from zirconium. So you don't see these signals. And we have plenty of examples that we have tried. We have tried room temperature. We have tried to collect 2K with helium. So we don't see it. So this is why I think they are complementary techniques. So it's always, if you want to study local structure, better to do PDF. If you are more curious about oxidation states, geometries, then XANES is maybe your technique.
Okay, thank you. Then I have another question here from Alejandro Fraccaroli from the organizing committee. Says, thank you very much for your presentation. I'm wondering if you can comment on the consequences of local zirconium clusters transition in the MOF bulk structures. So does it generate defects on the structure by disengaging organic linkers, or it's just variation on the distances of zirconium?
Sorry, I didn't quite get that. So what are the consequences?
The question is if it generates defects in the structure by disengaging organic linkers. So I'm thinking it's about the let's say the coordination of zirconium with the carboxylic ligand.
We don't think so, but this is something maybe we should. No, we don't think so. We don't think this is a mechanism. So we think it's a local distortion. We don't have missing linkers. We don't have different coordination of the linkers. Yeah, no, we don't think this is what is happening. But yeah, this is interesting. Yeah, because this is another local defect that is well described in metal organic frameworks. No, the ligand vacancy. So this is another kind of local defect, I would say.
So they say you don't generate this as coordinatively unsaturated metal sites, so you don't see that.
Yeah.
Good. Then another from our audience, Tamoy Mighty, he wants to congratulations on your good work, and he just want to ask if the palladium catalyst in this late slides and cuff, it's able to activate chloroarenes for Suzuki reaction or denitrification processes. So have you tried, or do you know if this
is something we haven't explored. No, we haven't explored, to be honest. So we did just a very easy carbon-carbon coupling because for us was just a proof of concept. But yeah, I mean, we are happy. We have the materials here. So if someone has ideas, we are happy to ship our materials. So no, we haven't explored because this is not our main focus, let's say.
Great. Then we have another one from Juan Alegreto. So thanking you for your presentation. And he would like to ask is there any lower limit on the signal noise ratio that you should be looking for in order to analyze the PDF signals in your experiment? So what is the lower limit? Let's say if you don't use synchrotron-based techniques, so you have in-house, let's say, X-ray sources.
So if you don't use synchrotron radiation, the problem is that you don't get what we call the Qmax. So for the people who are familiar with the technique, so you are very limited in the resolution of your data, the Qmax, okay? And that means that the peaks become really white, so you don't have these sharp peaks. And the second problem is that you have a lot of background. So these oscillations, the ripple terminations, which then you don't know what is a background, what is a signal. So this is why it's important to work with high energy radiation. I mean, if you have a diffractometer with a silver radiation, is a good option as well. So I know many cases if you can just run an experiment overnight, you know, if you don't want to do anything in situ, it's completely fine. And then then you can get decent data, but you cannot get good data with copper radiation, molybdenum radiation. I highly doubt. So yeah, this is why.
So how much is a typical collection time for synchrotron-based experiments in this PDF you show?
So it can go from 2 minutes up to 10 minutes. 2 minutes can be 5 minutes average.
Great.
Yeah, something fast.
Great.
Yeah.
Then Marcelo Sterini is thanking your comments for earlier question. And the last one from Jimena Tuninetti, he is also thanking you for your conference. And then she's wondering what possibilities do you have to measure a material such as SIG-8, which was reported to have an open structure as temperature effect when you go above 250. And in that case, what would you expect to see? A change in PDF peak area? How complicated it could be to compare structures of MOF with and without a loading, like a cargo molecule within the porous structure? So let's say you run it with some adsorbate in the porous structure.
No, this is actually, that's a good candidate for PDF analysis. So yeah, I don't know exactly what he wants to see. I didn't quite get the problem. So what kind of question is he trying to?
Yeah, it's like if you would like to analyze the, let's say, distribution of your cargo molecule within the MOF porosity by using different loading amounts by PDF.
Yeah, then with PDF, we cannot really determine the location of species. So for that, it's better to do low resolution density maps. This is something we did in Argon as well, but PDF is not really capable of determining the location of a species. What we can do with PDF, if you have a guessed species in the pores, we can determine the structure or possible interactions. That's something we can answer. So interactions of your guest molecules with a host, that's something we can do. Yeah.
Right. Also, you could see any distortions from
Yes.
the
Yeah,
structure with
distortions.
PDF.
Yeah, it's it. Yeah.
Right. Okay.
So,
I think we're almost on time. So maybe last one from me. I was I mean, I have no expertise on PDF, but I was wondering if it's not a chemical sensitive technique, right? Because you have to see interactions atom to atom in distance, like whatever geometry you're considering from your crystal structure. So how do you assign those peaks to a determined zirconium oxide or zirconium? So you use your crystal structure and you assign those
yeah,
peaks,
you
or
use your chemical intuition. So you need to have some kind of information. So PDF can get some chemical information if you do. Is a variation of the technique called partial PDF, in which you collect data close to the absorption edge of the element that you're interested in. So
right.
then the intensity of you really see variations in the correlations which are associated to this particular element. So something
Right.
is possible to do, but typically we have chemical intuition. So you know that it's a zirconium bond, so you know very well what are the range for the chemical bonds. Yeah, so you have some information.
Yeah. Okay. Yeah, that explained it. Very nice explanation, and thank
Yeah,
you
thank you.
again very much, Anna, for your excellent
Yeah, thank you so
exposition
much.
and
Mm.
your questions, answers, I mean. And we were looking forward to the next group papers from PDF in these iron clusters, which are really promising. And thank you again.
Yeah, thank you.
So